These are the learning objectives for section 4.4 on chromosomes and heredity. We'll go over them one by one as we get to them. So our first learning objective here is to describe the paired arrangement of chromosomes in human karyotypes. So we'll start with the definition of what heredity is. This is the transmission of genetic characteristics from parent to offspring. Here's another definition, karyotype. This is a chart of 46 chromosomes laid out in order by size. And so what they do there is they take a picture of chromosomes during metaphase of mitosis when the chromosomes are the densest. And then they cut out the pictures and then pair them up and order them from the biggest to the smallest. So we'll see a picture of one of those in a bit. The 46 chromosomes, they occur in 23 pairs. Each member of a pair is referred to as homologous chromosomes. Homo means the same. And you get one complete set from each parent. There are 22 of those pairs are called autosomes, and they look alike. Each pair looks alike, and each pair carries the same genes. There is one pair referred to as sex chromosomes because of the role they play in determining sex. If you're a female, you're going to have 2 X chromosomes, and if you're a male, you're going to get 1 X and 1 Y chromosome. This is what a karyotype looks like. You can see the chromosomes are ordered based on their size and their banding patterns. So the size and banding pattern and the location of the centromeres right here help to see which pairs are which. So this chromosome and this chromosome are carrying basically the same genes. Same with number 2 and so on. You can see some of the chromosomes have their centromeres off center. And here at the end, we see a case of what a karyotype would look like for a female, that last set of chromosomes, the X. And if this were male, then it would be 1 X and 1 Y chromosome. Here's another term called diploid. And diploid is going to describe cells that have both complete sets of chromosomes. And in all of your body cells, somatic means body, all of your body cells have both complete sets. So all your body cells are diploid. There's an exception. Those cells that are haploid, these are created with special cell division, which cuts the number of chromosomes in half. So basically, the complete sets are divided. And this is during the formation of sperm and egg cells. And again, the process is called meiosis, which is a special kind of division. Like mitosis for the body cells, when we're creating sex cells, we use the process called meiosis. Now, when a sperm and an egg cell unite during fertilization, they do form one single cell called a zygote. When the sperm and the egg unite, each one has one complete set. So when they unite, the zygote now restores that diploid number where you have 2 complete sets. So now we're on the second learning objective. The second learning objective says, let's define an allele and discuss how alleles affect traits of an individual. So first thing we're going to do is define the word locus. Locus is going to be a specific location for a gene on a chromosome. For example, a specific gene might be found on chromosome number 1. And the specific place where that gene is located on chromosome 1 is called its locus. And if you ever see the word loci, we're then referring to more than one location, which is also referring to more than one gene. Now, because of mutations that occur, sometimes you're going to have different forms of the same gene. So let's say we have, let's take something simple like determining flower color in a plant. And a flower has a red flower. or the plant has red flowers, and it just so happens that some individual plants in that same population, same species, have white flowers. So there you have two forms of a gene for flower color. One is red form, the other one is the blue form, and they would be referred to as these alleles for that specific gene. We also notice that there are alleles that dominate, or dominant alleles. And we're going to represent these symbolically with capital letters. So if a dominant allele is present, then you're going to end up seeing that trait in the individual regardless. And what this does is going to mask the effect if an individual also happens to inherit a recessive allele. Now, every individual is going to inherit, for most all genes, you're going to inherit two copies of the gene. And sometimes those two copies are the same allele, and you'd refer to as homozygous. And sometimes they're two different alleles, and one of the alleles happens to be recessive. So if you inherit the dominant and the recessive, then the recessive one gets masked by the dominant one. So let's say, in the example of the red flowers, if you were to breed a red flower, a pure breed red flower, which has both alleles red, and a purebred white flower, which has both copies of the gene or both alleles are white, and you breed them, and then you get red flowers in the next generation, all of them red, then you know that the red must be the dominant allele, because every one of the offspring plants had to have inherited one red allele and one white allele, because both of the parent plants were pure breeding, which means they had the same copies of both alleles. They were true breeding, they were homozygous for that. So these dominant alleles are often going to be responsible for any visible traits that you see, like for example in the flower color. And then we have the recessive allele. We're going to represent these with lowercase letter. And these traits are only going to be seen if an individual inherits both copies of the recessive allele. So we would say they would be homozygous for that. And many times this recessive allele is basically coding, it's a mutated gene, and it codes for a non-functional variant of a protein. So the protein doesn't function properly, and therefore whatever it's responsible for producing doesn't get expressed. So I have a picture here of two chromosomes, and on those chromosomes are specific locations, or loci, there you see the term loci, plural for locus, of three different genes. One gene we'll call gene P, another one gene A, and another one gene B. And we have two different colors here to represent that perhaps one chromosome was inherited from maternal and the other one from paternal during fertilization. So this is one pair of chromosomes, so the chromosomes have the same types of genes on there. But for gene P, this one seems to have inherited both capital letters, so we would say it's homozygous and it's dominant. This second gene, gene A, different location on the chromosome, is also homozygous. However, it's being symbolized with two small letters, so we're going to say that's recessive. And then an individual that has inherited one of each type of allele, we're going to call that heterozygous. So what I was referring to for an individual who inherits two copies of every gene is I'm referring to its genotype, and this is basically the alleles an individual possesses for a particular trait. So for example, in the first gene described on the previous slide, the individual had inherited both dominant alleles, so that would be considered its genotype for that particular gene. And so that's what we're referring to. So there's basically two categories of genotypes. You can be homozygous, where you get two identical copies of alleles, and it could be dominant or recessive. And then heterozygote individuals, where you get you inherit two different copies for a gene, two different alleles. Now, genotype is different than phenotype. Phenotype is referred to as a trait that you observe. So it's what an allele expresses, or what you see. For example, when I was referring to red and white flowers, those were specific phenotypes, and those phenotypes were determined by their genotypes, which was the set of alleles that they had gotten or inherited. Oftentimes, and I'm going to give an example here in a little bit, in order to analyze inheritance patterns and be able to make predictions, if you know the genotypes of parents when they produce offspring, when they mate. You can actually predict what the outcomes might be in terms of probability. So what are the chances of getting one genotype or even a phenotype once you know what their genotypes are. Now, the example the book gives you is going to be with a trait called cleft chin, and we'll see that here in a little bit. So it is a human trait. It's determined by one particular gene. But before that, we're going to go and look at flowers because it's easier to talk about flowers. So we're going to breed 2 flowers, and we're going to go with one red breeding with one white flower. And we're going to say, so I'm going to put an X there to say we're crossing them. And let's say that this one is true breeding, and this one is also true breeding, which means generation after generation we get nothing but red flowers when we breed them. Same thing for this, nothing but white flowers. Now we don't exactly know at this point, before we learn about it and make observations, we don't know which flower color is dominant. So when we cross them, that might help us. So we already know the history of each line. For the red, they keep producing red. For the white, they keep producing white. And if our results, when we cross these and make a hybrid, if our results give us red, then we can determine that the red allele was dominant. So now we'll assign a capital letter here, and we'll use R because red is the dominant. So we'll use capital letters here. We're going to use the same letter, but we're going to put lowercase letters, and we know they're homozygous because they were true breeding. So this gives us red, and we know that this must be heterozygous, and we get all red. Now, next thing we might want to do in a crossbreeding experiment is to get individuals from this generation. So get different individuals, get pollen from one plant, pollinate the flower of another, and here we would use a Punnett square analysis. So basically what we're going to be doing is we're going to now be crossing this second generation. And we're going to perform this cross, and we're going to use a Punnett square for our analysis. So this is our Punnett square. It's going to be a 2 by 2 square. And on top of each box, we're going to put the possible gametes that can be produced, the possible sperms and eggs, or pollen and ovules when we're dealing with plants. They can be produced during meiosis, whenever their gametes are formed. So we know that during meiosis we're going to be splitting up the chromosomes, which will split up the alleles. So this allele, the big R for this individual right here, the first individual, is going to end up producing half of their gametes with a dominant allele, and the other half are going to be with a recessive, so we can split them up here on the top of the box. So that's going to be one type of gamete. One out of the 2 boxes, or half of them, are going to be R, and the other half are going to be the recessive little r. And we do the same for the other parent, but we do it on the other side. And so we put a big R there and then a little r there. And now, let's say that this is a sperm or a pollen grain, and this is an egg or an ovule. And this pollen could possibly fertilize this particular, that's a possibility. And if you do that, when you combine the chromosomes, now you're going to have a nucleus that has those 2 letters. So it's going to be homozygous dominant. But it could be that this pollen grain here pollinates and combines with this ovule. And if you do that, then the nucleus would now contain this genotype, heterozygote. We can do the same thing for the bottom ovule. It could be this pollen grain during pollination combining with this, and so we get another heterozygote. Or it could be this pollen grain combining here and producing a homozygous recessive. So what is our end results here? We have 3 different genotypes. When we cross the hybrids, we have homozygous dominant, 1 out of 2. I mean 1 out of the 4 possibilities, or 25% chance. We have so this is going to be 1 out of 4. This is going to be 2 out of 4, or basically 1/2, 50% chance of getting heterozygote. And then we have a 1/4 chance here because only one of the boxes gives us homozygous recessive, so that's a 1/4 chance there. Now what we've done is we've given the probabilities of getting each of the genotypes, but what are the probabilities of getting red. These 3 boxes here would give us red. So there's a 3/4 chance of getting red when we're talking about phenotype. So 3/4 red. And we would see the white reappear 1/4 of the time in that second generation. What I've just described for you here is an inheritance pattern that would be referred to as complete dominance, in which one allele is completely dominant over another one. But let's say we take another red and white flower species. This is another kind of plant that happens to produce red flowers and also happens to produce true breeding white flowers. And again, this is another species of plant, totally different, but produces red and white flowers. And we cross them, and when we cross them, we end up getting not red flowers and not white flowers, but we get pink flowers. Here, it seems then that there's a blending of the 2 alleles. So we had a red allele and a white allele, and the heterozygote gives us a blending of it. So basically, if this were true breeding here, big R, and this were true breeding, homozygotes, and we get the heterozygote. So the heterozygote then would be a blending. That pattern is going to be referred to as incomplete dominance. Since no one allele is dominant over another one, so they're both kind of, it's a blending of each one. And now we can apply Punnett square analysis to this, and we can get our ratios here. The difference here is that we would get 1/4 red in a crossing like this. This other half, or 2 out of 4 that we were getting red earlier as a heterozygote, those would be pink, and then we would get 1/4 white if we were to run this analysis in incomplete dominance. So you would basically get 3 different phenotypes: red, pink, and white, as opposed to just red or white in complete dominance. And then let's say we come across another plant species in which we have true breeding red and true breeding white again, and when we cross them, we get flowers that produce on the same flower and the same plant. We get both red and white petals on there. So it seems that both in the heterozygote are equally expressed. So we get the red and the white. That would be referred to as wow, it escaped me right now. Oh, yeah, it would be referred to as codominance. I just had a brain freeze right there. Well, not a brain freeze. Well, you know what I had. So there you go. Those are some of the basic inheritance patterns. So in review, we talk about complete dominance here, where the heterozygote would look just like the dominant flower. In incomplete dominance, the heterozygote would be a blending of the 2 phenotypes, and in codominance, you're going to get both traits equally expressed. So let's move on to a case in humans of complete dominance, and this is a case of cleft chin. So if you don't know what cleft chin is, here's an example right here on John Travolta, one of those actors. He's got a cleft chin, and then here's Arnold Schwarzenegger, the Terminator, doesn't have a cleft chin. Okay, so the allele for cleft chin is dominant, and so you're going to get cleft chin if you are homozygous dominant and heterozygote, because it's complete dominance, and you would only get uncleft if you are homozygous recessive. So let's say we take a case and use a Punnett square where both parents are heterozygous. So for the mom, she's heterozygote, so big C and little c, so she can produce half of her eggs are the dominant allele, the other half are recessive. And then the male, the father, is going to have a big C here, and the other half of the sperm are going to be little c. And so we're going to get 1/4 homozygous dominant, 2/4 or 1/2 probability of getting heterozygote, and 1/4 chance of getting homozygous recessive with the uncleft chin. Now if we look at the phenotypes, we would get 75% chance of getting cleft chin though. So phenotype you would see 3 out of every 4 children born to parents who have those genotypes as having that cleft chin. So let's move on. So parents then can be healthy, but at the same time they could be heterozygote carriers of hereditary disease. So let's say we have a case where there's a allele that you have a gene, and the gene mutated, so you have an allele that's a faulty allele, and it creates a genetic disease or a genetic disorder. A parent could be carrying it, and I'll just use X here. If they're heterozygote and they're carrying the recessive allele for that genetic disorder, they would be perfectly normal, but there's a chance that their offspring is going to inherit that allele. And if they happen to marry someone and mate with someone that is a carrier as well, doesn't have the disorder, but they're carrying it, we know from the Punnett square analysis that there's a 1/4 chance that the individual would inherit that combination there and then have the genetic disorder. This brings up the idea of genetic counseling and genetic counselors. If you know you're an individual and you know you have a history in your family of genetic disorders, the same may be true of your partner or your spouse, and you want to plan to have children, you might go and see a counselor and see what the chances are of your offspring, your children, having this disorder, and then you can make some decisions about whether or not to have children or adopt or whatever the choices may be. Now we're going to talk about concept of multiple alleles, where this is a case where you have more than one type of allele for a gene, more than 2, not just 2 different alleles, but more than 2 alleles. So we'd call that multiple alleles. Another idea, codominance and incomplete dominance, which we already covered before, so we're familiar with those concepts. But if we look at a population of people, or a population of trees, or a population of flowers, or a population of dogs, basically there's going to be a gene pool. And this is the genetic makeup of the entire population. So basically there would be certain frequencies of alleles within that population. Now every individual has 2 copies of a gene. So every individual has 2 alleles. Now the alleles may be homos, they may be the same alleles, or they may be different alleles. But overall, if we looked at the entire population, there's a certain percentage of one allele and a certain percentage of another. So some alleles are going to be more common than others. And so we'll see what that can show us when we observe real populations, and the frequency with which we see certain traits in those populations. First of all, with multiple alleles, a good example for that is with humans. When we're dealing with the ABO blood types, there's basically 4 different phenotypes. You have blood type A, there's blood type B, there's blood type AB, and then there's type O. And there are 3 alleles for this. I'm going to symbolize this with a letter and a superscript. So there's the allele I with an A, so this is the A allele. Here's the B allele, and we use the capital letters because both of those are dominant. And they're dominant over allele with the symbol I, and I is for blood type O. So if we were to combine an A allele with an I or an O allele right here, then it would be blood type A because A is dominant over O. The same is true for B. Okay, so if we look at B here and we combine with O, then we would still be blood type B. So you could be blood type B if you have both B alleles, or if you're heterozygote for that. Now the exception here, or the difference here, so that's kind of complete dominance here. But if we were to combine these 2 alleles together in an individual, then both are dominant and they're codominant. So the individual would then be the blood type AB. And so you have, example here in this case, multiple alleles, and you have complete dominance, and you also have codominance, all in this cis blood type system that we find in humans. So it defines codominance is where both alleles are equally dominant. Both are phenotypically expressed. So an individual that is blood that gets the genotype AB, then they're going to be blood type AB. Then we have incomplete dominance, which we covered with the flower, where we got the pink flowers here. Heterozygote individuals show phenotype intermediate between traits of each allele. And there's an example from humans. This is similar to like the pink, the red and white producing pink flower. Here you have individuals that that have a phenotype where they usually have normal cholesterol levels in their blood, and then you have individuals who inherit a faulty gene, and it's a disorder that would create too much cholesterol in your blood, or hypocholesterolemia in your blood. Hyper means more, cholesterol, and emia means in the blood. And so in those individuals that have both copies, then they're going to have high cholesterol in their blood. And if you have one that's heterozygote, then they're going to be a shade in the middle, somewhere in between having normal and too much cholesterol. They're going to be somewhere in between. So that's the example of incomplete dominance when it comes to blood cholesterol. And then we have this concept of polygenic inheritance, and another one called pleiotropy or pleiotropy. Now, polygenic means more than one gene, and a lot of traits. This is why it's hard for me to come up with examples for humans, because those things we see, observe readily, like height, skin color, hair color, eye color, those types of things. Those things tend to be not very simple. Case of simple inheritance, one gene, one trait. Here, and the way you can tell is because that particular trait we're looking at, there's a whole range for that particular trait, a whole range of phenotypes, like a continuous range pretty much, where you can have all kinds of heights, for example, or all kinds of skin color, or all kinds of hair color and eye color. Like in this picture here, we have black, we have medium color here, a brownish color, and then we have no color at all, which actually gives us a blue color for eyes. But we have this whole range of color there. And so when you have that whole range, then you know it can't be just one gene. It's several genes that are responsible for producing that. And that's called polygenic. And if we did it, then we would only have basically tall and short. There wouldn't be anybody in the middle. So we know these are going to be polygenic. And the examples are many of the ones I gave there: eye color, skin color. But even some problems with cancer, some forms of cancer may be polygenic as well. And then we have pleiotropy, and this is where one gene produces multiple phenotypic effects. And the example given in the book here, so you have one gene, and then you can see many effects here. The example is actually a disorder called alkaptonuria, and this disorder results from a mutation on chromosome 3, which blocks the breakdown of tyrosine. Basically, it creates a faulty enzyme in the metabolism of breaking down tyrosine. And so tyrosine is transformed to another compound, and then that compound is supposed to go through another change, but it can't because the enzyme is not produced correctly. So it stays there, and then that particular compound goes and starts combining with some of the collagen fibers and connective tissues and so on, and then it reacts with oxygen and produces blue color. And so you'll get darkened skin, you'll get darkened whites of the eyes or the sclera here, and your urine, because this compound will come out in your urine, again, when it's exposed to oxygen, it'll turn this dark color, which is kind of weird and crazy. So that's an example of pleiotropy, where you see all of these different effects as a result of one gene. Then we have sex-linked traits, and sex-linked traits are sex-linked because they occur on the sex chromosome. So here in a female has 2 X chromosomes, but a male only has 1 X chromosome, and then they have a smaller Y chromosome. So it's hypothesized that this chromosome actually used to be exactly homologous to the other X chromosome, but over time in males, it started losing bits and pieces. So basically what we've done is we've lost many of the genes on this Y chromosome. So really, they're only going to get 1 copy of the genes from the X, whereas females will get 2 copies. So here, basically a male, for any genes that are on the parts that no longer exist on that chromosome, they only get 1 copy. whereas females get 2 copies. Well, it just so happens that some traits are linked to this X chromosome, and they're referred to as sex-linked traits. There are a few traits linked to the Y chromosome, but many of the ones we know come from the bigger X chromosome, which has many more genes on it. So here there is a particular it's a genetic disorder where a gene involved in whether you can see color or not. We have the normal copy here, and then we have it mutated, and so this individual female is heterozygote for that. The male can't be heterozygote or homozygote because they only carry 1 copy. So the female, while she has 1 bad copy here, she has 1 good copy, so she's not going to have the problem. So she's going to have normal vision. But if a male, which can only inherit 1 X chromosome, let's say this is the mom, she could either give this allele or this allele to her offspring. If she gives this one and it's a male, well, the only way he can get the X from is from the mom. Then he's going to end up getting that disorder. So you tend to see these sex-linked traits, you tend to see them more common. You're going to see the problem in males more. So you'll see like this color blindness. If you ask around, you know anybody who's colorblind, and then they do, and you ask them, well, what's their gender, what's their sex, and so they're male, so it's most likely male. Now it's possible for a female to be colorblind, but she would have to have a father that's colorblind and a mother that's carrying, and then she would have basically half a chance. If she's a girl, she would have half a chance, or 50% chance. So we ran a Punnett square and then analyzed it. She would have 50% chance of getting a color blindness, but the chances of getting a colorblind male and a female who's carrying it is very low. So you'll see less females who are colorblind in this sex-linked, particular sex-linked traits such as these. There's others too, like hemophilia. There's some of the hemophilia can be traced to the X chromosome, so it's sex-linked. So males tend to have problems if they inherit the bad copy. They'll get in situations where they can't stop bleeding, so that they get that hemophilia disorder. Here's another concept now and a new objective. So our new objective is to discuss the interactions of heredity and the environment in producing individual traits. We have something called penetrance and another one called environmental effects. So for penetrance of an allele, there's going to be a percentage of a population that shows or exhibits an expected phenotype. For example, we ran a Punnett square and we knew what an individual should show, what phenotype. We might not necessarily get that within the population when we look at the entire gene pool. Okay, so just because the individuals actually may have those particular alleles, they don't show it. And a good example here is we don't see the kind of frequency we should for polydactyly. And polydactyly is where individuals are born with an extra digit, like an extra finger or extra toe. And here the penetrance is only about 80%. So individuals that actually have this, and it's a dominant allele, individuals that end up inheriting it, only about 80% will show. So the penetrance is 80%. And then we have environmental factors here that can influence gene expression. An example here is from production of melanin here, and it's diagrammed over here. So in order to produce melanin, which gives us our skin color and our eye color and our hair color, we need to consume in our diet an amino acid called phenylalanine, because we cannot produce that one ourselves. So we can make many amino acids, but there's some we can't, so they're essential and we need to get them in our diet. And it turns out we have a couple of genes, and those genes are responsible for, after we go through transcribing and then translating, they produce these enzymes. There's 2 enzymes needed in this metabolic pathway. The phenylalanine would be acted on by the first enzyme, and change it to tyrosine, and then the second enzyme would change tyrosine to the melanin. But what happens when you don't get phenylalanine in your diet? Then you can't produce the tyrosine, then you can't produce the melanin. So That's an environmental effect, something from the environment. In this case, your diet. You're not able to get enough phenylalanine in your diet. So now this dominant and recessive alleles, when it comes to gene pools, we talked about the number of alleles total that are out there in the population. So every allele that you can count, and there's 2 per individual, right? So if you have 100 individuals and each one is carrying 2 alleles for each gene, then there is a total of, for any gene, there is a total of 200 alleles in that population. So this brings up here a common misconception that dominant alleles must be more common in a gene pool than recessive ones. And so this is not true. Just because a allele is dominant doesn't mean you're going to see it all the time. Okay? And some recessive alleles, like blood type O, so we're now looking at the other side here, some recessive alleles are actually going to be more common. So again, it's opposite of what you think. Because it's recessive, you would see it less frequent in the population. But just because it's recessive doesn't mean a lot of individuals have those alleles. That there's many of them carrying even both copies, right? So then you would tend to see it more in the population. And some dominant alleles, like polydactyly and blood type AB, are rare. And these are basically due to dominant alleles. So these are specific examples where just because polydactyly is dominant, you don't see a whole lot of extra digits within the population. And just because blood type AB is A and B are dominant, you don't see a lot of AB people with AB. There's a smaller percentage in the population. And then this brings up now, let's go back here to epigenetics. And epigenetics is a field that examines non-genetic, this is relatively new area where learning more and more about it. But here, non-genetic changes that can alter gene expression. So here you're not actually changing genes, you're not mutating genes, but certain changes on the DNA itself without changing the sequence pattern in the DNA, certain changes that occur with the entire, that occur there, other than changing the sequence within the DNA, can actually have and be passed on to offspring. So this is kind of interesting. Here, that basically you alter something about the chromosome, and it does not allow the expression of a gene, or it might allow the expression of a gene more often than it should. And this is without a mutation in the sequence, as I mentioned a while ago. Now, one way this can happen is with a process called DNA methylation. This is one of the mechanisms for epigenetic change, and this is where a methyl group, now remember methyl groups from our study of chemistry, here it's a CH3, okay? So that's added to the DNA, and this methylation actually silences the gene. And unfortunately, sometimes this inappropriate DNA methylation may be responsible for silencing some genes responsible for suppressing cancers, right? So remember, there's those oncogenes. Well, I think we're going to cover that in a little bit. Oncogenes, and then there's suppressor genes that actually work against uncontrolled growth of cells or cancer. So this is the case here. Now, this is not in your book, but I went in and looked it up, or a picture to show how this methylation occurs. Well, here we have a piece of a nucleotide that is, and it's usually one of the nitrogenous bases that's going to be methylated. So here is the way the base looks normally. I believe it's C, or the cytosine. And here we're basically going to replace, there's a hydrogen right here. It's understood that there's a hydrogen right there. And we're going to replace that hydrogen and put the methyl on there. And here you can see these familiar structures here. Remember, these are, each of these where the DNA is wrapped around 1.5 times is called a nucleosome there. And so when the methylation occurs in this, along the DNA strand, that methylation is going to occur on the C base, or the on the cytosine. And here's another change called acetylation. So methylation would work to suppress the gene. and acetylation would work to make that particular gene transcribable. So when it's acetylated, it's open to expressing, but if it's methylated, it would be turned off. Now this methylation can actually be passed on. There you go with epigenetic change that reduces the transcription of certain genes, basically silencing them. So now we come up with the idea of cancer. I think I started mentioning it a little while ago, but we'll get to what I was trying to talk about. And here is a diagram of a tumor, and it's malignant. And what makes it malignant is that it's fast growing and it metastasizes. And metastasize means that cells will break off and spread. So they'll travel through your bloodstream, or they'll travel through your lymph vessels, and then they'll end up leaving those vessels and migrating to some other location, and then beginning to grow there. And so then you have a secondary tumor that started from the primary tumor where the initial cancer had started. And cancer is, just by definition, the uncontrolled growth of these cells. Now we might distinguish this from benign tumors, which are encapsulated and don't tend to metastasize. And those types of tumors could be removed very easily without having major problems. So this the study of cancer is called oncology, and so this is a study dealing with tumors. And here's a concept here that the tumors actually are going to need blood supply. They're growing rapidly. A rapid mass of cells are going to need a blood supply. They're going to need a nutrient supply. They're going to need to be able to get rid of the waste they produce. So these tumors tend to have what's called tumor angiogenesis. Angio is going to be referred to blood vessel, and genesis to produce. So definition here is growth of blood vessels by these energy-hungry tumors. Cancers are named for the tissue of origin. For example, carcinomas are of epithelial tissue. Lymphomas are of lymph nodes. Melanomas are going to be cancers of pigment cells in the epidermis called melanocytes, the ones that are responsible for giving you skin color. Leukemias are going to be in blood-forming tissue, where white blood cells are produced, and then sarcomas are going to be in bone or other connective tissue or muscle. Now there are agents in the environment that do cause or are cancer causing, so you're exposed to them, your risk of getting cancer is greater. So they're called carcinogens. One of them is radiation, including ultraviolet rays from the sun. This is where skin cancer is a problem. X-rays are a problem, or can be a problem. If you get too much X-rays, that could be an issue. There are chemical agents like cigarettes, the tar from cigarettes, food preservatives, like in those preserved meats, the nitrates, they get converted into carcinogen once they hit your stomach acid. And then lots of industrial chemicals are known carcinogens. And then there's viruses. Being infected by a virus can also lead to cancer. There's the HPV, or human papillomavirus. There's hepatitis C, and then there's type 2 herpes simplex, which are known to be associated with causing cancer. Only about 5 to 10% of cancers are hereditary. And carcinogens, what they do is they trigger gene mutations. Those gene mutations then later result in the cancers. So we're going to look at some specific genes associated with cancer. There's one called oncogenes. These cause cell division to accelerate out of control. And the way these types of genes do it is they're going to produce, the genes are responsible for accelerating the production of growth factors, which get cells to go through division, or they accelerate the production of growth factor receptors on the membranes of cells. And so there's more interaction with those growth factors with those cells. And then we have another group of genes called tumor suppressor genes. So healthy tumor suppressor genes are going to inhibit the development of cancer. Some of them code for DNA repair enzymes, and a mutated tumor suppression gene, or a silenced tumor suppression gene, will leave those oncogenes that we talked about a while ago here unstopped or unopposed. So basically these are the tumor suppressors are kind of like the brakes, like they put the brakes on for the oncogenes. to help balance. So if we do have mutations or carcinogens affecting these tumor suppressors, then that could be a problem. This is a picture of a tumor in the kidney of a child. It's called Wilms tumor, and there you can see this mass of cells right here in the middle of the kidney, really getting into the kidney and basically destroying the tissue within the kidney and robbing it of nutrients and so on. So pretty sad condition, I think. So what are the lethal effects of cancer? How is it that cancer can be a problem? So here is one. The growing cells are going to replace functional tissue and vital organs. They're often going to invade blood vessels, lung tissue, or brain tissue, which are all vital organs. Cancer tumors are going to steal nutrients from the rest of the body, and they're going to cause a condition called cachexia, which is severe wasting away of depleted tissues because the tumor is just taking up all of the nutrients, so the other tissues nearby are wasting away from that. Cancers can also weaken one's immunity, and that can leave the door open for infection, opportunistic infections.